Hello, Namaste. Welcome to Shah's Biotech for you. Today we'll continue with the tools of biotechnology. The second tool is cloning vector. Cloning vector is a DNA molecule into which a foreign DNA can be inserted or integrated. This can self-replicate within the host cell to produce multiple copies or clones of recombinant DNA. Coming to the features of cloning vector, the first one is ORI. ORI stands for origin of replication. It is a DNA sequence at which replication is initiated on a chromosome, plasmid, or virus. ORI, a cloning vector should possess an ORI for it to self-replicate inside the host cell. The second feature is recognition site. This is the DNA sequence which is recognized by the restriction enzyme and it can be cut. This facilitates the insertion of the target DNA. Third is selectable marker. Selectable marker facilitates the screening of the recombinant organisms. Example is antibiotic resistance gene. A cloning vector should be of small size, that is less than 10 kb in size, for easy integration into the host cell. It should possess multiple cloning sites. It should be capable of large DNA insertion and to work under both prokaryotic and eukaryotic systems. This is the simple pictorial representation of a cloning vector which has an ORI, selectable marker and a multiple cloning site. Let me discuss about the significance of multiple cloning sites with an example. If the multiple cloning site has ampicillin resistance gene and tetracycline resistance gene, then on ligating a foreign DNA in the recognition site within the tetracycline sequence, it will obviously disturb the DNA sequence. Hence, the recombinants or the vectors with the DNA with the foreign DNA will lose the tetracycline resistance. But this cloning vector will have ampicillin resistance gene intact. Hence, on introducing the mixture of both recombinants and the normal cells into a medium with ampicillin and into another medium with tetracycline, the recombinants will grow in ampicillin but will not grow in tetracycline as they have lost its resistance due to the insertion of foreign DNA. But non-recombinants will grow both in ampicillin and tetracycline containing medium. This phenomenon of losing the resistance because of the insertion of a foreign DNA is called as insertional inactivation. Hence, the presence of MCS or the multiple cloning site here helped us to differentiate between recombinants and the non-recombinants in a visible manner. Now coming to the types of cloning vectors. Plasmids, bacteriophage, phagemids, bacterial artificial chromosomes, yeast artificial chromosomes, cosmids, retroviral vectors, human artificial chromosomes are the types of cloning vectors. With this, I wrap up today's session.